Urinary tract infections are the second most common bacterial infection worldwide. Pathogenic E. coli are the major causative agents of these infections. Cranberry has been thought to aid or protect against UTIs. Research has shown that cranberry-containing PACs reduce the movement of E. coli by affecting flagella production. PACs seem to directly reduce the expression of fliC, which codes for the flagellar protein flagellin. Previous research that we looked at used pathogenic E. coli, but for ours, we wanted to use a non-pathogenic strain called K12. We had started with the primers used on the pathogenic strain in other research, but soon realized that it does not work as well with our strain. In order to get the proper primers, we had to put the strain through amplification and send it out in order to have the primers produced for us. In the bottom left, you can see our forward and our reverse. And that was able to give us the blue and yellow code needed in order to have the primers work for our K12 strain. With our GAP-A primers, they actually ended up working pretty well with our strain. We don't have them exactly where we want them to be, but we are planning on coming back to them at a later date. But for our qPCR reaction that we do, it does seem to work well as a reference gene and it doesn't change the expression. And then here we've got our melt curves. We have fliC and we have GAP-A. So what this does for us is it actually tells us that all of the amplicon DNA is all melting at the same temperature, which does prove that we have from blue sequence to blue sequence right above, and nothing else that would be affecting our qPCR results. So then for our production of cranberry extract, we mixed freeze-dried cranberry powder with sterile water and then mixed them further for 30 minutes and centrifuged for 10 minutes. We then decanted the supernatant, pH adjusted the mix and filtered it through cheesecloth before putting it through a sterile 0.2 micrometer filter. This was able to give us a proper cranberry count in each one. We then took RNA from the cells and ran them through an RNA extraction kit, which is listed below in here. And that was able to, so we used their method in order to get the RNA that we needed, which we then exposed to DNase and then reverse transcribed into cDNA in order to put it through qPCR. So up here, we used, in order to analyze our qPCR results, we used the common base method, which was developed by my advisor. This method allows for us to do a regression relating the changes in cranberry to the changes in fliC expression. So on the graph, you can see for 5 versus 0, the expression of fliC is 87%, that to 100 and 0. And for 10 versus 0, the expression is lower at 75% for 10 to 100 at 0. The line is inverse, but as we increase cranberry, we do see a decrease in fliC expression. And for E. coli and cranberry, we wanted to test if cranberry's effect on fliC expression was direct or indirect. This experiment showed us that the growth of E. coli in the different cranberry concentrations didn't change and grew just as well with any concentration of cranberry in it. This actually suggests to us that cranberry has a direct mechanism effect on fliC expression and is not just causing E. coli to divert resources for survival. And currently, we are also running tests on agar plates with different cranberry concentrations in order to see the movement of E. coli and how it is affected at each concentration of cranberry.